So we finished going over the different components of the immune system: innate, adaptive, the different cells that go along each, how they interact with each other, and just the overall scheme. And now we are going to talk about colonial selection and expansion first, and then we're going to move on to hematopoiesis. So let's first start off with colonial selection. So colonial selection is a principle of the adaptive immunity. So colonial selection is really just a characteristic of adaptive immunity. It's basically saying that every B cell and T cell have specific receptors, and how these receptors only recognize and bind to very specific antigens. So basically, when a pathogen enters the body, only the lymphocytes, B and T cells that have the receptor for that pathogen will be activated. So it's not activating the entire immune system every time one little pathogen enters the body. And so this just ensures that there is a targeted immune response and not your whole body freaking out. So we can kind of write that down in bullet points. So adaptive immunity, we can say that B and T cells have unique receptors. And then we can say when a pathogen enters, only specific lymphocytes, B and T cells, get activated. And the overall goal of this is just to ensure a targeted immune response. This is basically just no unnecessary activation. Okay, that is colonial selection. Now we're going to move on to colonial expansion. So colonial expansion is basically saying that when a B cell or a T cell is activated, when it binds to an antigen, it rapidly proliferates. So it just multiplies itself over and over again so that there's just a large population of the same identical cell to fight that pathogen that has entered the body. So for B cells, for example, it would differentiate into either plasma cells or those memory B cells, which we talked about in the last video. And then for T cells, it could also differentiate into different effector T cells, like helper T cells or cytotoxic T cells, which we talked about in the last video, or it could also turn into memory T cells. So again, we can write down this in little bullet points so that we can remember. We can say after B or T cell binding, rapid proliferation. And so this rapid proliferation is really technically the colonial expansion. And so this is going to make a large population of identical cells. And so this is basically just so that trying to make it more effective to fight this infection. And so I'll just quickly note that those B cells can go into plasma or memory, which we already talked about. And then those T cells can go into helper, cytotoxic, or even just like memory T cells. So that is pretty much everything for colonial selection and expansion. Now we are going to move on to hematopoiesis, which is the formation of these different immune cells. So I'm going to draw a cell just right here in the middle where all of our other immune cells are going to come from. And this cell is called a hematopoietic stem cell. And from here, it can go two ways. It can go the myeloid lineage or the lymphoid lineage. And so one is going to be innate immunity and one is going to be adaptive. So let's start with the myeloid lineage. The myeloid lineage is going to lead us to a myeloid progenitor, and then the lymphoid lineage is going to do the same thing, but instead it's going to lead us to a lymphoid progenitor. All right, let's start with the myeloid lineage, starting from this precursor. Our myeloid cell can differentiate into 6 different cells in the innate immunity pathway. So first is something called a monocyte, and the next is something called a eosinophil, and then we go into our neutrophils, and then we have our mast cell, and then we have our basophil. And then from here, the monocyte specifically can differentiate into 2 different other very important cells that we have heard about before, the macrophage and the dendritic cell. Okay, and then we can quickly just write down the function of each one of these cells. So the monocyte obviously is just kind of circulating in the blood, and then when it reaches a pathogen or needs to differentiate for phagocytosis, it's going to differentiate into either a macrophage or a dendritic cell. Now, eosinophils, on the other hand, these target allergic reactions and also parasites. Neutrophils are phagocytosis, which we talked about earlier, and then mast cells, these release histamines, and histamines have to do with that inflammation response, and they also play a big role in kind of like wound healing, we can put. And basophils similarly also produce histamines for that allergic and inflammation response. And then we can scroll down here to the macrophage and dendritic cell, and we did already go over these in the last video, but just quickly, these ones, phagocytosis, and remember they activate T cells with that MHC2, and then the dendritic cells, remember, are also antigen presenting cells with that MHC2, and these were the bridge between adaptive and innate. Okay. So that is all of the cells for the myeloid lineage for the innate immunity. Now we are going to move over to the lymphoid lineage. So instead of differentiating into 5 different cells like the myeloid one did, this one is only going to be differentiating into 3. We have the T cell, the natural killer cell, and then we have the B cell. And then from here, if we remember, these T cells can differentiate into helper T, cytotoxic T, and then those memory T cells. And then the B cells, if you also remember, can differentiate into those plasma cells, and then those memory B cells. All right, and then we can just quickly label what each one of these do. This T cell differentiates into all of these 3 different kinds. The helper activates B cells, cytotoxic kill those other cells, and then those memory T cells are for long term. And by long term, I just mean that they store memory so that next time the same pathogen comes along, we can fight it more efficiently because we already know what to do. Next, these natural killer cells basically just recognize either missing or abnormal MHC1. And so these are actually a part of the innate immunity, even though they come from the lymphoid lineage, because they are not specific. What makes them different from like cytotoxic T cells, for example, is they act immediately. They don't need antigen presentation on MHC1 to target it for attack. It just recognizes anything at all that's a little bit off and then just automatically attacks it. So because it is not specific in that way with the antigen binding, it is a part of innate immunity, even though it's from this lineage. Now these B cells, if we remember, they produce antibodies. These are what binds to those antigens to signal the innate cells to come over and kill it. Plasma cells, again, produce antibodies, and then those memory B cells for long term. Okay, so that is everything for hematopoiesis, which just basically states that all of these immune cells stem from this one cell and how they go through different lineages to get through all the different cells that we see in our immune system. We can zoom out and kind of see how everything fits together. We have our overall pathway, colonial selection and expansion, which we talked about in this video, and then everything from the last video, just about the different branches of the immune system, how they interact. But that is all I have for the immune system notes. The completed notes will be on the link tree. This will be under biology, and then it will be just the immune system is what the file will be called. And that is all I have for you today.